Hello everyone, welcome to this video. My name is Priyanka. In this video, we are going to discuss about isolation of RNA by Trizol method. Aim: to isolate RNA from cells by Trizol method. Principle: the isolation of RNA with high quality is a crucial step required to perform various molecular biology experiments. Trizol reagent is a ready to use reagent used for RNA isolation from cells, and Trizol works by maintaining RNA integrity during tissue homogenization, while on the other side it also helps in disrupting and breaking down cells and cell components. Addition of chloroform after the centrifugation separates the solution into aqueous and organic phase. RNA remains only in the aqueous phase. After transferring the aqueous phase, RNA can be removed by precipitating with isopropanol, but the DNA and proteins can be recovered by sequential separation after the removal of aqueous phase. Precipitation with ethanol removes DNA from the interface, and an additional precipitation with isopropanol removes proteins from the organic phase. Total RNA extracted by Trizol reagent is free from the contamination of proteins and DNA. This RNA can be used in Northern blotting analysis, in vitro translation, poly A selection, RNA protection assay, and molecular cloning. In this picture, we can see that the RNA remains in upper aqueous phase, DNA in interphase, and proteins in lower organic phase. RNA is removed by precipitation with isopropanol. Materials required or reagents used in this process are Trizol reagent, 0.8 m sodium citrate or 1.2 m NaCl, isopropanol, chloroform, 75% ethanol in DEPCH2O, RNase free water. Instruments that we use in this experiment are centrifuge and an incubator. Steps involved in this process: it contains 5 steps: cell lysis, phase separation, RNA precipitation, RNA wash, redissolving of RNA. First step: cell lysis or homogenization. Growth medium on the cell was discarded and cells were washed with ice cold PBS buffer. The monolayer was then covered with 1 ml of Trizol, and the cells were lysed and homogenized by repeatedly pipetting. Second step: phase separation. The homogenized samples were incubated for 5 minutes at 15 to 30 degrees for the complete dissociation of nucleoprotein complexes. 0.2 ml of chloroform per 0.75 ml of Trizol reagent was added. The tubes were shaken vigorously by hand for few minutes and incubate them at 15 to 30 degrees for 2 minutes. The sample were centrifuged for 15 minutes at 12,000 rpm at 4 degrees centigrade. The aqueous phase was transferred to other tubes. Following centrifugation, the mixture separates into a lower red phenol chloroform phase, an interphase, and a colorless upper aqueous phase. RNA remains only in aqueous phase. The volume of aqueous phase is about 70% of volume of Trizol reagent used for homogenization. RNA precipitation. The RNA was precipitated from the aqueous phase by mixing with 3 microliters of glycol and 500 microliters of isopropanol. The mixture was centrifuged for 30 minutes at 12,000 rpm at 2 to 8 degree temperature, and the RNA precipitated from a gel-like pellet on the side of the tube at bottom. Fourth step is RNA wash. The supernatant was removed. The RNA pellet was washed once with 75% ethanol, adding 900 microliters of 75% ethanol per 0.75 ml of Trizol reagent used for the initial homogenization. The sample were inverted and mixed and centrifuged at 12,000 rpm for 30 minutes at 4 degrees centigrade. Fifth step, redissolving RNA. The RNA pellet was dried. RNA was dissolved in RNase free water by passing the solution through pipette tip for a few times and incubating for 10 minutes at 55 to 60 degrees. The steps, once we recall it, preparing sample for which RNA should be isolated. The sample is treated with Trizol. After centrifugation, 3 layers are formed. Upper colorless aqueous phase contains RNA, interphase contains DNA, lower organic phase contains proteins. Second step, phase separation, in which the sample is treated with chloroform. This helps in partition of RNA into aqueous supernatant for separation. Third step, RNA precipitation. In this, the sample is treated with isopropanol. After centrifugation, pellet is formed. In the last step, the pellet is washed with RNase free water or TE buffer. Expected results: a gel-like pellet on the side and bottom of the tube of RNA precipitate. On taking OD at 260 nanometers and 280 nanometers, the A260 by A280 ratio should be above 1.6. Quality of RNA: the RNAs is 80 to 85% present, which can be visualized because they are rRNAs. rRNA should form two sharp bands in ethidium bromide gels. mRNA can be visualized because it is negligible. And A260 by A280 ratio should be close to 18 to 20. If the ratio is close to 20, when the isolated RNA is from low amount of RNA sample. Nanodrop is advanced spectrophotometer which requires very less amount of sample. Example, 1 ml sample is required for analyzing the RNA quality in this type of spectrophotometers. Value less than 1 indicates contamination with salts or phenol or proteins in the RNA solution. Thank you. Hope you like this video.